Welcome, I'm Dr. Jeff Borenstein. Everyone is touched by psychiatric conditions, either themselves or a loved one. Do not suffer in silence. With help, there is hope. Today, on Healthy Minds.
And I thought, what's the most important problem in all of psychoanalysis? It's memory. We are who we are, in large part of what we learn, what we remember. So we're beginning to fit several aspects of, mother's teaching about how to age successfully into a biological perspective.
That's today on Healthy Minds. Healthy Minds is brought to you in part by the American Psychiatric Association Foundation, the Graham Beck Foundation, the New York State Psychiatric Association, and the New York State Office of Mental Health. Welcome to Healthy Minds. I'm Dr. Jeff Borenstein. Today I speak with Dr. Eric Kandel, whose discoveries earned him the Nobel Prize in Medicine for his research on the molecular and cellular mechanisms underlying memory. Dr. Kandel shares his most recent findings about age-associated memory loss and steps you can take to keep your brain healthy and your memory intact. He also describes research, including discoveries which may lead to new treatments for post-traumatic stress. In addition, Dr. Kandel shares his personal experiences, which led to his extraordinary career researching the brain. Eric, welcome. Thank you for joining us again.
Jeff, I'm delighted to be here as always.
I want to begin by asking you, what got you involved in understanding memory?
It's a fascinating question. I was born in Vienna, Austria, and when I was 8.5 years old, Hitler marched in and changed my life forever. My friends stopped talking to me. I was beaten up in the park. Kristallnacht, everything of value was taken away from us. We were able to leave because we had a relative in the United States. But that experience forever left in my mind sort of the theme of the Holocaust. Never forget. It really is like my life has been sort of a post-traumatic stress disorder recovery, if you will. Trying to come to grips with this. How could people, educated people, listen to Haydn, Mozart, and Beethoven one day and beat up the Jews the next? In America, in school, particularly in high school at Erasmus, I became very much interested in history, and I continued that at Harvard. I majored in history and literature. And then through a friend, I got to know Ernst Chris, a very well-known psychoanalyst. And he said, if you want to understand the human mind, you're not going to do it through intellectual history. You've got to do it by studying the mind through psychoanalysis. So I decided I would go to medical school, and on short notice I took pre-med courses and went to summer school between my junior and senior year, was accepted to medical school, and went to medical school with the idea of becoming a psychoanalyst. In my senior year in medical school, I thought that even a psychoanalyst should know something about the brain. And in those days, unlike today, there were very few labs working on the brain, but there was a very good one here at Columbia, Harry Grinfest, and I worked with him, and I'd never had an experience like this before. I thought science was boring, but working in the lab, doing experiments, planning them, the sensual feeling of carrying out, doing things with your own hands was just wonderful. And I remember dating Denise, whom I later married, saying to her, I could see doing this for a living, but it's unrealistic. You don't have any money. I don't have any money. You want to get married? And she said, money is of no significance. I must say, in the 60 years since then, she's not repeated that.
She hasn't said that afterwards.
But anyway, it inspired me to go on. And because of that experience, Grinfest recommended me to the NIH. And Wade Marshall, who was the head of my lab, said, what problem do you want to work on? And I thought, what's the most important problem in all of psychoanalysis? It's memory. We are who we are, in large part of what we learn, what we remember. And also memory is so important in terms of my own experience. So that got me going on the problem of memory. And first I started studying memory in a very complex system, the hippocampus, because we knew it was important in memory. But I realized this was 1957, this a little bit too early. I mean, we wrote, Walden Spencer and I wrote 4 beautiful papers that got a lot of attention. but it told us a lot about how the cells in the hippocampus worked. But we didn't have a system that was modified by behavior. So I realized one had to take a radical reductionist approach. And I looked around for an appropriate system. I focused on a marine snail aplysia because it has a limited number of neurons that are gigantic. So since my specialty was recording from individual nerve cells, which very few people were doing in those days, having big cells was wonderful. Our lab was the first one to demonstrate that the connections between nerve cells are not fixed, but are plastic. They change with experience. In short-term memory, they undergo a functional change. They're just more effective, but anatomically, they're normal. But with long-term memory, you actually see a growth of new synaptic connections.
So the brain actually changes?
Absolutely changes. And at that time, I wrote a paper called Psychotherapy in the Single Synapse, based on a book that Helen Gage and Brown, I think, had written at that time called Sex in a Single Girl. And I pointed out that insofar as psychotherapy works, it must produce anatomical changes because it's a learning experience. And Helen Berryberg has since then shown that. That made me realize that this is a very useful approach, and I began to study the molecular underpinnings, what gives rise to the growth of new synaptic connections. And that's what I've really done for the last 30 years. And then with the advent of mice, I began to study not only normal memory, but also began to study disorders of memory. And one of the things I've become interested in recently is difference between age-related memory loss and Alzheimer's disease.
And there's a big difference between that. Tell us about that differentiation.
When I was a medical student in the 1950s, we practically never spoke of Alzheimer's disease. I mean, we knew it existed, but we didn't discuss it much. Why is that so? Well, people didn't live long enough to have Alzheimer's disease. In 1900, and we were seeing people born at that time, the average lifespan was 50 years. In the year 2000, the average lifespan for men is 78 and for women is 82. A major increase in lifespan. And Alzheimer's, you see it rarely in the late 60s. You begin to see 2 or 3% of people in their 70s. By 90s, half the population has Alzheimer's disease, so it's an epidemic. But as people lived longer, they also lived better. And one began to see, something that wasn't clear. It wasn't clear whether it was an early stage of Alzheimer's or whether it was a separate process, just like your body weakens with age and your muscles get a little bit stiffer. Maybe there are some changes that occur in the brain that accompany this, that are age-related memory loss, not Alzheimer's disease. And so my colleagues and I began to explore this. One simple experiment, which I did quite early, is to ask that question in mice. Mice do not show spontaneous Alzheimer's disease. So if you put a gene in it, they'll show Alzheimer's disease, but on their own, they never show it. So I asked the question, do they show an age-related memory loss? Sure enough, they get to midlife, fine, the memory is perfect. After that, it declines. And that's what's thought to be the case with age-related memory loss, benign senescent forgetfulness. I then began a very productive collaboration with Scott Small. He had done some imaging experiments, and he had found that Alzheimer's disease, which we knew begins in the entorhinal cortex, that age-related memory loss seems to begin in the dentate gyrus, different region of the brain. We took about 50 brains of people varying in age from 40 to 95, which did not have Alzheimer's disease. And we compared the entorhinal cortex of the dentate gyrus to see whether there's some specific genes that are expressed in the dentate gyrus that vary systematically as a function of age, either going down or going up. And we found 19 candidates. We're beginning to explore a bunch of them, but the first one we studied in detail is something called RBAB48. It goes down systematically as a function of age. Now, why is that interesting? It turns out RBAB48 is part of the switch that converts short-term to long-term memory. My lab had shown that that switch involves a transcriptional activation, so you turn on gene expression, and it's mediated by CREB, called the cyclic AMP response element binding protein. CREB binds to another protein called the CREB binding protein, but the machine that really makes the thing work is RBAB48. So there are 3 proteins that hang together. So part of the critical machinery for converting short-term to long-term memory in a normal circumstance decreases with age. And we found that RBAB48 decreases only in the dentate gyrus systematically as a function of age. Doesn't do it in any other region. And now we could do some nice experiments. We could take a young mouse and we could knock out RBAB48. We showed that now it acted like an old mouse, severe age-related memory loss. We could take an old mouse and restore RBAB48 and we could rescue age-related memory loss.
So with those two steps, you prove that causal relationship.
In the mouse.
Yes.
Yes, yes. And it looks, since we know that those molecules exist in people and people have age-related memory loss, it's a likely candidate. And then by accident, we learned something very exciting. And that is, I have a colleague across the street, Gerard Kasenki, chairman of the genetics department, and he's been interested in bone. What could be more boring than bone? And he's found that bone is an endocrine organ. It releases a hormone called osteocalcin. And osteocalcin acts on the gonads, it acts on the liver, it acts on the pancreas, acts on many things. And he found it gets into the brain and acts on the brain. So we began to ask the question, does it act on RBAB48? Does it act on the dentate gyrus? Does it improve age-related memory loss? And the answer is it does. If you give osteocalcin, it acts on RBAB48. It enhances age-related memory loss. If you knock out RBAB48, that has no effect. So this is very nice. Moreover, we found that as the animal ages, its bone mass decreases and osteocalcin decreases, okay? And that accompanies age-related memory loss. Now, if you exercise the animal, the bone mass increases, you release more osteocalcin, and you improve age-related memory loss. So we're beginning to fit several aspects of, mothers teaching about how to age successfully into a biological perspective.
Understanding a mechanism of action of the benefits of exercise for the aging brain.
Perfectly put. Yes.
Well, I know that you exercise on a regular basis.
I do indeed.
Tennis and swimming.
I'm going to go swimming after this is over.
So how does this research relate to what you see as future discoveries that can have an impact upon people's aging brain?
I think there are probably many, many factors that contribute to age-related memory loss, and we're identifying several of them. Cognitive involvement is also very important. How that works, we don't know yet, but there's no reason why you can't do this in mice. Ask them to solve different learning tasks and seeing whether this affects osteocalcin, whether this affects bone mass, whether it acts through some other pathways, which it may very well act on.
So exercising the brain.
Exercising the brain in a different way than just
physical
exercise. So there are lots of things to be done there. So I think, very worthwhile things to follow up on. Another thing you may be interested in is that with these animal models, we've also become interested in other psychiatric disorders. Eleanor Simpson in my lab has developed a very interesting model on schizophrenia. But what I really want to talk about is a more recent model we've developed on post-traumatic stress disorder. It's quite fascinating. In the course of studying memory, we made a very interesting discovery, first in a snail and then in a mouse. When you form a long-term memory, you activate CREB, you grow new synaptic connections. That keeps the memory going for several days. But then you need a process to maintain it beyond it. And the way we found this is if you apply inhibitor protein synthesis to the synapse, memory collapses after 48 hours, after 2 days. So we realized that local protein synthesis at the synapse, which people had known about for years, has a function in maintaining memory. So we asked, what regulates local protein synthesis? And we found that it's a molecule called CPEB, cytoplasmic polyadenylation element binding protein. Carl-6C was in my lab at the time, and he began to explore this, and he found that this was a very interesting protein. It acted like a prion protein. Now, prion had been very well described by Stanley Pusner. They're killers. They're responsible for Jakob Creutzfeldt disease. They're responsible, one thinks, for Alzheimer's disease, for Parkinson's disease, for Huntington's disease, protein folding disorders. These protein exists in two forms, a single soluble form and an aggregated form. The aggregated form forms for reasons one doesn't know, spontaneously, but once it forms, it becomes self-perpetuating, it kills the cell, is released from that cell, taken up by other cells, kills those cells, and this is how, like an infection, it spreads from cell to cell. And Stan Prions studied this, got the Nobel Prize for that. We found a prion that was different. When it moved into its aggregated self-perpetuating form, that was its normal function. So this was the first discovery of a functional prion in brain. It had been suspected from work done a couple of years earlier in yeasts, but no one had looked at this in complex organisms, no one had looked at it in the brain. And we thought this is fascinating. We found it first in the snail, then we found it in the mouse. You knock it out, perfectly good long-term memory, but it's not maintained. I told the people in my lab, one functional prion is a biological curiosity. Two is a biological principle. Let's go out and find another example. So Joe Raymond in my lab began to sort of look around, and he was struck there was a protein in many cells of the body called TIA, T-cell intracellular antigen, that's involved in cellular stress. So he became interested in knowing, is it critical for systemic stress? Could it be involved in post-traumatic stress disorder? So he looked at post-traumatic stress disorder in wild-type animals and animals in which TIA is knocked out. And he found that if you knock TIA out, the animals that have the knockout show dramatic enhancement to response of stress. But they only do this in female mice. It's sex-specific. Male mice do not show any change with the knockout. And we found this is a sex-specific glucocorticoid receptor splicing factor. And we presume there's going to be a sex-specific for males as well, and we're looking for that. But what's interesting about this is now allows us to study in detail how this works. We have an example of a second prion. We've put this into yeast. It has all the characteristics of a prion. But post-traumatic stress disorder is very interesting because it's associated with a number of correlated disturbances. And that is, if you smoke or if you drink, you're more susceptible to post-traumatic stress disorder. And if you have post-traumatic stress disorder, you're more likely to take up smoking or drinking if you haven't done it before. Now, we've been studying smoking and drinking because my wife is interested in how kids get involved with drugs. And she developed the gateway hypothesis in a very famous science paper in the early 1970s.
Which explains how one drug leads to another.
Exactly. And she showed that kids don't start with cocaine or heroin. They start with smoking or drinking, and then go on to these other drugs. But the question is this just a correlation or is it causative? So she looked around for a collaborator and didn't find anyone. Then she realized she's sleeping with somebody who works with mice, and she talked me into this. And we began to collaborate. And we showed that if animal takes nicotine and then cocaine, nicotine dramatically enhances the effect of cocaine. If you give the mouse cocaine and then nicotine, zero effect. Moreover, we showed that, and we showed this on the behavioral level, on the level of physiology, and level of gene expression. A very interesting story on gene expression. Moreover, we were able to show that the enhancement of cocaine by nicotine requires that the animal have nicotine on board when it tries cocaine. If you separate it by a week, nicotine has no effect. She went back and reanalyzed the epidemiological data, and it's exactly the way it is. We now have similar data for alcohol. Alcohol enhances cocaine. Cocaine has practically no effect on alcohol. So this is really quite interesting, and now it allows us to see whether or not these processes interact with post-traumatic stress disorder. This could be really quite fascinating.
Given the enormity of the problem of post-traumatic stress.
Enormous problem.
And that we still need much improved treatment.
For people like us who are interested in mental health, this is a major problem. Because it not only occurs in the military, it occurs throughout life. I think every Holocaust survivor, and what I experienced is mild, My wife was in hiding for 2 years. We all, to some degree, suffer from this.
And now, given some of the problems going on currently in the world,
enormous.
Think of how many people are affected.
Enormous, enormous.
Ultimately, what type of treatment would you envision that leading to for post-traumatic stress?
Well, I mean, we're now seeing how you can really, think of several ways. One is we're finding drugs that disaggregate these aggregates. So one way is to give a drug that causes these prions to fall apart. Okay. Second thing is to sort of define some of the molecular pathways in greater detail and affect them, just in a very specific fashion. And finally, the psychotherapeutic things. I'm a big friend of psychotherapy. I think the problem with psychoanalysis is not that it is not interesting or that it does not work, but that people have not taken the effort, or at least didn't when the time was ripe, to show whether it works and under what circumstance and how it works.
How it works.
These are important questions. I mean, Aaron Beck showed this for cognitive behavior therapy. People are beginning to show this for insight therapy. So I think, this is very likely to be extremely useful. And people do find it beneficial, but we'd like to have better documentation for that. And there's desensitization therapy of various kinds, on which people are exposed to the same experience without any fear associated with it, so they desensitize to it, if you will. So I think there are a number of behavioral therapies that could be very, very useful here. I'm high on behavioral therapy.
And sometimes a combination of potentially medication and
Absolutely.
Other interventions.
Yes, yes.
I want to shift gears back to the age-associated memory loss issue and ask you, what do you envision as potential treatment that may result from the work that you've been doing?
Well, I think it would not be very difficult, since we know the pathways, a cyclic AMP-dependent protein kinase, that acts on CREB and the CREB binding, to be able to reverse age-related memory loss. We can do it in mice very nicely. So I would think that this would be possible. And it becomes more realistic now. Until quite recently, the FDA did not distinguish between age-related memory loss and Alzheimer's disease. So every drug that was developed had to be tried on Alzheimer's. Now this is going to have no effect on Alzheimer's whatsoever, okay? But if one now can define a separate population that has age-related memory loss, I think trying these drugs is going to be very interesting. And I think several companies will do that.
So similar to how we now are proactive to help maintain heart health with treating cholesterol, treating hypertension, as well as exercise, diet, there may be medications as well as other steps people can
Absolutely.
take to protect
the obvious
their brain.
steps, what your mother tells you, is stay in good physical health. Don't overeat, keep your weight down, make sure your lipids are under control, make sure your blood pressure is under control, make sure your diabetes, if you have any, is controlled. That's number one. Number two, stay physically active. Go for walks, very healthy exercise. Swim if you like it, play tennis. But walking is a key to people as they age, because some may not be able to play tennis or may not enjoy it, may not have a pool accessible. Walking is accessible to everybody, or a treadmill or something like that. Stay socially engaged. This is why very important for people as they begin to have hearing deficits to get themselves hearing aids. People are very reluctant to get hearing aids because they think it makes them look old. But they don't realize that if they have difficulty following conversations in a group, after a while they drop out of the conversation. And after a while it becomes a habit to drop out of conversation. And that isolates them socially. And that's neither enjoyable nor good. So I think being socially involved on a variety of levels is critical.
Very good
And
point.
learning new things,
Take up a new hobby, a new interest.
a language, whatever.
All excellent advice. I want to thank you so much for again joining us.
Jeff, it's a pleasure.
Good. Thank you so much. We just heard how scientific discovery can sometimes be like a puzzle with different pieces coming together. Understanding what happens in the brain to cause age-associated memory loss can help us develop new interventions to maintain a healthy brain. For example, we know that exercise, both physical and mental exercise, can help keep your brain healthy. Staying socially active and engaged are also important for brain health. Until next time, I'm Dr. Jeff Borenstein. Goodbye. With help, there is hope. Healthy Minds is brought to you in part by the American Psychiatric Association Foundation, the Graham Beck Foundation, the New York State Psychiatric Association, and the New York State Office of Mental Health.